So welcome to this Pharmaceutical Technology Thought Leadership Series broadcast. I'm Rita Peters, Editorial Director of Pharmaceutical Technology. In this conversation, Brent Moody, Principal Scientist, Catalent Pharma Solutions, will talk about ways drug companies can assess the stability of a formulation in the early development phases. So welcome, Brent.
Thank you, Rita.
What is a stability indicating method?
Well, a lot of times our clients will come to us with analytical methods that they have been developed by their drug substance manufacturers. And in those cases, the drug substance vendors are usually focusing on determining potency, but also evaluating purity in the form of process impurities that are in their final product that they're coming out the door with. They're not necessarily looking at degradant-related species. So a truly stability indicating method is one that is specific for degradant-related substances, and that's important for formulation development because once we as formulators receive the compound and start working on it, it's going to be exposed to environmental stresses. And unless you have confidence that the analytical method you're using to assess your prototype formulations and ultimately your final formulations are going to be able to see, separate and quantitate those degradant species, you could potentially run into problems. So what we want to see is an analytical method that has been demonstrated to be stability indicating by force degrading a compound through ICH defined conditions of acid, base, heat, light, and peroxide. And to do this, we want real life degradation, so we force degrade it to about 5 to 15%. Beyond that, you begin to try to separate degradants of degradants. So we'll do that, and we'll modify the LC conditions to achieve separation, and we demonstrate peak purity by photodiode array. We look for baseline separation, and ultimately we like to see some semblance of mass balance. And at that point, we feel confident that we have a basic liquid chromatography method around which to build the quality control methods of our drug product, where we're going to assess in process manufacturing. We're going to look at release testing and ultimately support stability studies for shelf life.
Why is a stability indicating method important in early phase formulation development?
Well, I sometimes like to say that doing formulation development without a stability indicating method is kind of like driving a car in the dark with no headlights. You can have the best platform technologies that do a great job of enhancing your solubility, but if you're only looking at potency, you're only looking at the solubility of your compound, you're not looking at keeping an eye on the compound stability through the compounding process, the formulation process, and ultimately these drug products have to sit on a pharmacist's shelf. So you have to have a good stability indicating method to be able to assess that and make informed decisions about what is the right platform for you to go forward.
So what tools does Catalent offer to improve the method development?
Well, ultra high pressure liquid chromatography is a tool that we use. It's been around for a while, but it was a little slow to be accepted by the industry. I think some of that was led by not all quality control labs had UPLC capabilities, but Catalent has been really on the forefront of putting that into our laboratories, and I think over the past couple of years there's been a lot more acceptance of it. That allows us to develop methods and run methods a lot faster. We get much sharper separation, better accuracy in our results, and perhaps most importantly, we use a lot less harmful analytical solvents. So that's good for the environment and the community as well. So we're proud to be on the forefront of that.